In this screencast, I would like to cover the following learning outcomes. Please note that some of the topic I'm going to cover today are linked to the content delivered by Dr. Black in week 20 in the lecture titled Introduction to Blood Systems. So please do refer to Dr. Black's lecture for further details on some of the topic I will cover today. How is oxygen transported in the blood and within our bodies? Well, between 1.5 to 2% is dissolved in plasma. The rest is reversibly bound to hemoglobin inside the red blood cells, erythrocytes. Hemoglobin is a quaternary structured protein composed of 4 subunits arranged in a ring-like fashion, as shown in this cartoon here. In this case, hemoglobin is composed by 2 alpha chain and 2 beta chain, with an iron atom covalently bound to the heme in the center of each subunit. And the heme is responsible for binding the oxygen. One hemoglobin molecule contains 4 iron-containing heme molecules. Therefore, each hemoglobin molecule can carry up to 4 molecules of oxygen. Oxygen binds to hemoglobin in a cooperative binding or in a cooperative way. What does it mean? It means that the binding of the first oxygen molecule causes a conformational change in the hemoglobin that allows the second molecule of oxygen to bind more readily. And as each molecule of oxygen is bound, it further facilitates the binding of next molecule until all 4 heme sites are occupied by oxygen. And when all 4 heme sites are occupied, the hemoglobin is said to be 100% saturated. And in a healthy individual with normal hemoglobin level, hemoglobin saturation generally ranges from 95 to 99%. You have different kind of hemoglobins. Hemoglobin that is binding to or is bound to oxygen is called oxyhemoglobin. Unbound hemoglobin is called deoxyhemoglobin. You have a type of hemoglobin called methemoglobin, which is a form of hemoglobin in which the ion in the heme group is in a ferric state and not in a ferrous state of normal hemoglobin. The problem is that methemoglobin cannot bind oxygen, and the presence of methemoglobin obviously results in a impaired transportation of oxygen in diseases. Can be inherited, but also can be caused by exposure to certain drugs, chemicals, and even food. Another type of hemoglobin is the fetal hemoglobin, where the beta chain has been is substituted with a gamma chain, which has got a higher affinity for oxygen compared to the regular hemoglobin. Finally, you have the hemoglobin S, which presents an abnormal beta chain, which results in a lower affinity of hemoglobin for oxygen. And the presence of hemoglobin S is responsible for the sickle cell disease. How is carbon dioxide transported in the blood? Well, between 5 to 7% is dissolved in plasma. The rest is actually entering the red blood cells and can either bind the hemoglobin, and the carbon dioxide can bind hemoglobin at different side than oxygen. And as you will learn later on in more detail, the binding of carbon dioxide to hemoglobin reduces the affinity of oxygen for hemoglobin. However, the majority of carbon dioxide is actually carried in the blood as bicarbonate. Now, how is this bicarbonate formed? Once carbon dioxide enters the blood cells, can react with water to form a weak carbonic acid, and this reaction is catalyzed by carbonic anhydrase. Carbonic acid is an unstable molecule that dissociates into bicarbonate, ion, and hydrogen. Since the carbon dioxide is quickly converted in biocarbonate ions, the reaction allows for a continuous uptake of carbon dioxide into the blood down to its concentration gradient. Obviously, you will note that there is a production of hydrogen in here, and if too much hydrogen is produced, it can alter the blood pH. However, hemoglobin can bind the free hydrogen ion and can limit the shift of the pH to a more acidic value. What happened to the biocarbonate ion, which is newly synthesized? Well, it is transported outside the red blood cells in the plasma and is exchanged for a chloride ion. This is called chloride shift. However, in the lungs, what happens is that the bicarbonate is reconverted, thanks to the carbonic anhydrase, to carbon dioxide, which is exhaled. Now, before describing this slide and the oxygen hemoglobin dissociation curve, I like to remind you few major points that will help you to understand the dissociation curve. So remember that gases travel from an area of higher partial pressure to an area of lower partial pressure. Remember that oxygen is able to combine in a reversible manner, to bind in a reversible manner with hemoglobin. Remember that the affinity of an oxygen molecule for heme increases as more oxygen molecules are bound to the hemoglobin. Is a cooperative binding. And remember that each hemoglobin can bind a maximum of 4 oxygen molecule, and in this configuration the hemoglobin is said to be 100% saturated. If 3 molecules are bound, the hemoglobin is said to be 75% saturated. 2 molecules bound, 50% saturation. 1 molecule of oxygen bound to hemoglobin is said to be 25% saturated. So now, why do we have an oxygen hemoglobin dissociation curve? We use this curve because it describes the relationship of partial pressure to the binding or dissociation of oxygen to heme. And it tells you that the percentage saturation of hemoglobin is a function of the partial pressure of oxygen. Basically, partial pressure of oxygen, so this PO2 here, determines the degree, the amount of binding of oxygen to hemoglobin at the side of respiratory membrane, and obviously, in the other end, the degree of dissociation of hemoglobin of oxygen from hemoglobin in the tissue. Now, this curve is characterized by a sigmoidal shape, and this tells us that the percentage saturation of heme does not increase in a linear fashion as the partial pressure of oxygen increases. So what you can see is that there is a very steep saturation increase of hemoglobin when the partial pressure of oxygen increases from 0 to 4. So there is the saturation increases very quickly. And why? Well, that's due to the fact that the binding of the first molecule of oxygen to heme will increase the affinity for the second molecule, and so on. As mentioned earlier, there is a positive cooperativity in the type of binding. The sigmoidal shape of the curve explains why oxygen is loaded into pulmonary capillaries, and unloaded from systemic capillaries into the tissue. So in tissue, you have a lower partial pressure of oxygen, and the affinity of oxygen for hemoglobin is decreased. So the unload of oxygen to tissues is facilitated. On the other end, in the lungs, in the alveoli, where the partial pressure of oxygen is high, there is positive cooperativity, don't forget that. The affinity for oxygen, of hemoglobin for oxygen is high, so the loading of oxygen is facilitated. What else can we say when we look at this curve? Well, we can see that there is a flat portion of the curve between 60 and 100 of partial pressure of oxygen, which is a positive thing in the sense that because of that, humans can tolerate a decrease in alveolar partial pressure for oxygen, say due to changes in atmospheric pressure, without compromising the amount of oxygen that can be transported by hemoglobin. Now, this curve can change, and changes in partial pressure of carbon dioxide, pH, temperature of this 2,3-diphosphoglycerate affect the dissociation curve, affects the affinity of hemoglobin for oxygen. And this factor, and we will talk about that in more detail in a second, can shift the curve to the right, here in purple, or to the left, here in blue. And as I said, the shift of the curve to the right or to the left will reflect the changes in the affinity of hemoglobin for oxygen, not in binding capacity. Right, so a shift to the right of the dissociation curve is occurring when there is a decrease in affinity of hemoglobin for oxygen. So shown in here in purple. And it's caused by an increase of CO2 and therefore an increase in partial pressure of carbon dioxide, or in a decrease of pH. So acidity, an increase in acidity can cause a shift to the right. An increase in temperature or an increase on these 2,3-diphosphoglycerate or 2,3-DPG can also cause an increase, a shift to the right of the dissociation curve. Just a few words about this 2,3-DPG. This is an intermediate of glycolysis in erythrocyte, which is rapidly used under normal oxygen tension, so normal oxygen concentration. However, if or when hypoxia occur, so there is a reduction of oxygen in the peripheral tissue, the concentration of this intermediate of the glycolysis can accumulate to significant level within hours. And at this concentration, 2,3-DPG can bind to the deoxyhemoglobin, and by doing so reduces the affinity of hemoglobin to oxygen. That's why this results in a shift to the right of the oxygen hemoglobin dissociation curve. And obviously this facilitates the release or the unload of oxygen to tissue encountering long-term hypoxia. Anyway, going back to the causes that are responsible for the shift to the right of this curve. The increase of partial pressure of CO2 and the decrease of pH are and the impact of this factor on the dissociation curve are described as Bohr effect. So the Bohr effect is the effect caused by a increase of CO2 or a decrease in pH on the oxygen hemoglobin dissociation curve, which leads to an increased unload of oxygen to the tissue. What you also have is that there is an increase on this P50. You might hear of that. This is the partial pressure of oxygen at which hemoglobin is 50% saturated. So in this case, if we are looking at the normal condition, and let's look at the green trace in this case, when hemoglobin is 50% saturated at a value of 25 a partial pressure of 25. Okay, so if the dissociation curve is shifted to the right, as you can see, there will be an increase of the partial pressure of oxygen at which hemoglobin is 50% saturated. Indeed, you will have 50% saturation of hemoglobin this time, and now look at the purple trace now, around a value that is higher than 40 millimeter of mercury. Obviously, you can, if you have a decrease in the partial pressure of oxygen and an increase of pH, a decrease in temperature and decrease of 2,3-diphosphoGlycerate, you can have an increase in affinity of hemoglobin for oxygen, and then therefore you will have a shift to the left of this dissociation curve, shown in here as in light blue. And obviously, you will have a decrease in the P50, so a decrease in the partial pressure of oxygen at which hemoglobin is 50% saturated. So if you look at the curve, at the blue curve in here, you have that at the partial pressure of oxygen of 20 rather than 25, 30, the oxygen is 50, the hemoglobin is 50% saturated. Now, when do we have, for example, a shift to the right of the oxygen hemoglobin dissociation curve? Well, we have that when we have an increase in metabolic activity in tissues. So when we do exercise, during exercise, the increased metabolic activity in tissue causes an increase in CO2, therefore an increase in partial pressure of carbon dioxide. You will have formation of carbonic acid and an increase on hydrogen, therefore a decrease in blood pH. Increased metabolic activity in tissue also increase lactic acid, that can lead to a decrease in blood pH. And the heat produced by working muscles will increase the body temperature. These are all factors that can promote the shift to the right of the oxygen hemoglobin dissociation curve. Why? Because then more oxygen can be delivered to hard working muscles. You have certain adaptation in certain condition, for example, at high altitude, that impact, if you like, on the dissociation curve, or on factors that impact on the oxygen hemoglobin dissociation curve. So at high altitude, you have adaptation that include the increase in blood cells number and an increase in hemoglobin, but also an increase in synthesis of the 2,3-DPG. Now, if you remember, DPG was produced during hypoxia. Well, it's always produced, but during hypoxia can accumulate. Now, during in a high altitude, or living at high altitude, can cause will lead to hypoxia. Because of that, there is a stimulation of the production of 2,3-DPG in red blood cells. There will be an increase of 2,3-DPG, which will facilitate the delivery of oxygen to tissue. And this is an adaptive mechanism, as it is also the increase of angiogenesis. In the last couple of slides, I would like to talk to you about the changes that we have in gas exchange and also in oxygen and hemoglobin dissociation curve during pregnancy. Now, during pregnancy, the fetus has his own circulation. and erythrocytes, but obviously depends on the mother for oxygen. So the blood supplies, if you like, the sequence of supplies is shown in here. So you have the maternal vessels, and then the placenta, and the fetal chorion, and then the umbilical vessels. So you will have umbilical vein and arteries. The gas exchange occurs at chorion, similarly to gas exchange at the respiratory membrane in our lungs. However, there is a reduced diffusion of oxygen. Why? Because the partial pressure in maternal blood is roughly between 35 to 50 millimeter of mercury, and the partial pressure of oxygen in fetal blood is around 20 millimeter of mercury. So you understand that the difference is not as large as it is in our lungs. If you remember, in our lungs, in the alveoli, the partial pressure of oxygen is around 104, and in the capillaries around 40 millimeter of mercury. So the differences between the maternal blood and the fetal blood is not large. So there is not as much diffusion of oxygen into the fetal, in fetus. So adaptations are obviously necessary to ensure a good oxygen supply to fetus. So which are these adaptation? Well, first of all, we mentioned the fetal hemoglobin earlier on, and I mentioned that the fetal hemoglobin has got a gamma chain rather than a beta chain, and has got a greater affinity for oxygen when compared to the adult hemoglobin, or the normal hemoglobin. Second, the fetal blood has a very high red blood cells count compared to maternal blood. So more red blood cells there to pick up oxygen. Last but not least, in fetus, we have what is called double Bohr's effect. Why we have the Bohr effect occurring twice? Well, because when the fetal blood loses CO2, its pH is increasing. And if you remember, an increase in pH increases the affinity of fetal hemoglobin for oxygen. When maternal blood gains CO2, the maternal blood will show a lower pH, and therefore the hemoglobin in the maternal blood will have a reduced affinity for oxygen.